Okay, so let's get started. So good morning and good evening, everyone. So welcome to, I think, this year's last Immuseum seminar. It's really my pleasure to welcome today's guest speaker from Japan for the very big ending of 2022. So welcome to Dr. Kiyoshi Takeda. So Kiyoshi is currently a professor in Department of Microbiology and Immunology Graduate School of Medicine in Osaka University. At the same time, he is also a director in Osaka University Immunology Frontier Research Center. So very, very brief background introduction about Kiyoshi. So Kiyoshi actually graduated from Osaka University School of Medicine back in 1992 and conducted his PhD work at the Graduate School of Medicine, also in Osaka University, under supervision of Professor Shizuo Akira, who is really famous with a lot of innate immune regulation biology. So Kiyoshi became an assistant professor in Kyoto College of Medicine and Research Institute of Microbial Diseases, Osaka University, where he worked on the mechanism for toll-like receptor-dependent pathogen recognition. In 2003, he became a professor at the Medical Institute of Bioregulation, Kyushu University, and then moved to Graduate School of Medicine, Osaka University in 2007. Currently, as I just mentioned, he is a director and professor of Immunology Frontier Research Center in Osaka University. So his lab mainly focus on understanding the pathogenesis of inflammatory bowel diseases, particularly the analysis on how intestinal homeostasis is maintained by mucosa innate immune cells and epithelial cells. In particular, his lab is interested in several different directions related to the GI homeostasis, which including the intestinal immunity, which I just mentioned. They try to characterize the unique function and regulatory machinery of innate myeloid cells in the intestinal lamina propria. Second part, his lab also working in the intestinal environmental factors, which they try to analyze the effect of microbiota and the dietary components on the host immunity. Third part is his lab working on the intestinal epithelial cells, try to analyze barrier function of a single layer in the epithelial cell, in the intestinal epithelial cells, which play a very important role for burial integrity, intestinal immunity, and then microenvironment factors. But as also said, besides that, his lab also tried to analyze how microbiome residence in the intestine tissue without evoking inflammatory responses and influence on the host's health. So as I just mentioned that Kiyoshi actually really make a lot of groundbreaking contribution to the intestinal biology and then microbiome community, particularly with his work a couple of years ago on the new factor LYPD8, regulating the intestinal homeostasis. And I think today we're going to hear him talking about host and microbiome interaction for the maintenance in the intestinal homeostasis. So thank you so much, Kiyoshi, for doing this for us, for the audience. And we are very much looking forward to your talk. Please go ahead.
Okay, thank you, Chuan. So really nice introduction. And I'd like to appreciate Chuan and Charlie for providing the opportunity to speak in this seminar, Zoom seminar. So as the Chuan kindly introduced, our laboratory is interested in the pathogenesis of the inflammatory bowel diseases represented by the ulcerative colitis and Crohn's diseases. And recent evidence have shown that both genetic predisposition of the intestinal immunity and intestinal microbiota both contribute the pathogenesis of the inflammatory bowel diseases. So actually the complicated interaction between the intestinal immunity and microbiota leads to the development of the inflammatory bowel diseases. But between this intestinal immunity and microbiota, there exists a single layer of the intestinal epithelial cells. So we just wondered if these epithelial cells have some barrier function to separate the microbiota from the intestinal immunity. In this respect, epithelial cells of the gastrointestinal tract are covered by the mucus layers. And compared with the epithelial cells of the stomach and the small intestine, epithelial cells of the colon, where the tremendous number of the microbiota do exist, these colonic epithelial cells are covered by the very thick mucus layers composed of the outer mucus layer and this inner mucus layer. And then when we stain this microbiota and this mucus of the mouse colon, this red stained microbiota are really abundant in these outer mucus layer regions. But when you see this inner mucus layer regions, which is visualized by these MAC2 mucins, these inner mucus layer regions are essentially free of the microbiota. Thus, by the presence of this inner mucus layer, microbiota are completely separated from the host cells. And for the mechanisms for the separation of the intestinal microbiota and the host cells, so general concept so far accepted is that because this mucus is really slimy, this slimy mucus mechanically prevents the invasion of the bacteria. But we just wondered if this is only one mechanism. So I asked one of our PhD students to analyze the another functions. He supposed that a molecule that is selectively expressed in the epithelial cells of the colon might be responsible for these functions. So he searched for the molecules that is selectively expressed in the colonic epithelial cells. And he found this molecule named LYPD8. When we analyzed mRNA expression of this LYPD8, this LYPD8 mRNA was expressed only in the gastrointestinal tract, such as stomach, small intestine, cecum, and the large intestine, particularly in the cecum and the large intestine. And when in the large intestine, LYPD8 is selectively expressed in the epithelial cells, but not in the lamina propria. So LYPD8 is selectively expressed in the epithelial cells of the large intestine and cecum. So this is just expression of mRNA. So next generated monoclonal antibody, and he analyzed protein expression. So mouse colon was fixed with this Carnot's fixation method and used for the immunohistochemistry. And in this case, LYPD8 was found to be highly expressed in the uppermost layer of these colonic epithelial cell regions. In addition, LYPD8 was found to be secreted into the intestinal lumen. To visualize the outer mucus layer and the inner mucus layer, microbiota was also stained. In this case, LYPD8 was found to be accumulated into this interface of the outer and inner mucus layers. Thus, LYPD8 is secreted and accumulated into this interface of the outer and inner mucus layers. So this is the case for the mouse. We then analyze the human samples. In the case of the human samples, colon was just fixed in the classical PFA fixation method. In this case, the luminal contents were all washed out, but in this case, red stained LYPD8 was again highly expressed in the uppermost layer of the colonic regions. When we analyze the ulcerative colitis patients, LYPD8 protein expression was severely reduced. Thus, the ulcerative colitis pathogenesis might be correlating with the expression of the LYPD8. So we then analyzed the role of LYPD8 in the colon by going back to the mouse system, and we generated LYPD8 knockout animals. In the case of the wild type animals, again, we can see the bacteria-free region just above the colonic epithelial layers. But in the absence of the LYPD8, although in this area, we can have the thick mucus layers, but let's stain bacteria attach or invade into the colonic epithelial cells. So we then analyzed what kind of the bacteria invade into the colonic tissues. So DNA was isolated from the colonic tissues and analyzed by the qPCR using several bacteria genera specific primers. Actually, in the LYPD8 knockout colonic tissues, several bacteria were increased, particularly Proteus, Escherichia, Helicobacter were tremendously increased. These bacterial species are all the flagellated gram-negative bacteria, which they show the high motile activity. We also took another approach. Colonic tissues were homogenized and just cultured on the LB agar. In this case, in the knockout LYPD8 knockout tissues, the number of the bacterial colonies was increased. In addition to the typical bacterial colony, we can see the very unique colonies that show the swarming migration patterns. DNA sequencing analyzing of this colony have shown that this bacterium is the Proteus mirabilis. Several reports have shown that the number of Proteus mirabilis is increased in the inflammatory bowel diseases patients. We then analyzed the sensitivity to the intestinal inflammation of these LYPD8 knockout animals. So we already treated with the small amounts of the dextran sodium sulfate to induce the severely mild inflammation in the wild type animals. Under these conditions, LYPD8 developed very severe intestinal inflammation, and many LYPD8 knockout animals was dying. Thus, LYPD8 was shown to be highly sensitive to the intestinal inflammation. So, we then wondered if this high sensitivity to the intestinal inflammation correlates with the disappearance of the bacterial-free space just above the colonic epithelial regions. To address this, mice were then orally treated with the two kinds of the antibiotics, gentamicin or the vancomycin. Gentamicin is active for the gram-negative bacteria, including Proteus mirabilis. In contrast, vancomycin was active for the gram-positive cocci, but not for the gram-negative bacteria. Oral treatment of the vancomycin did not induce anything in the LYPD8 knockout animals, but oral treatment of the gentamicin reduced the number of the gram-negative bacteria in the intestinal lumen and accordingly restored these bacteria-free regions. So we then analyzed this sensitivity to the intestinal inflammation of this gentamicin treated LYPD8 knockout animals. So again, LYPD8 knockout, this red line, LYPD knockout animals were highly sensitive to the DSS induced intestinal inflammation, and they showed severe weight loss. But after gentamicin treatment, LYPD8 knockout animals became resistant to the intestinal inflammation. So, in the absence of the LYPD8 gram-negative bacteria showing the high motile activity, invade into the colonic tissues, and this increased the risk of the intestinal inflammation. Even under these conditions, gentamicin-mediated elimination of this gram-negative bacteria restored the bacteria-free region, and they became resistant to the intestinal inflammation. indicating that LYPD8 is involved in the maintenance of the intestinal homeostasis. So we next analyzed how LYPD8 inhibits invasion of these gram-negative bacteria to the epithelial layers. We just analyzed whether LYPD8 binds to the fecal microbiota. Fecal microbiota were incubated with a recombinant LYPD8 protein, and the LYPD8 binding was analyzed by the flow cytometry. So actually, LYPD8 binds to some microbiota. Because this is the flow cytometry, we isolated this LYPD8 bound microbiota, and DNA sequencing have shown that these bacteria are the Escherichia or the Helicobacter gram-negative bacteria. So we then analyzed which portion of the bacteria LYPD8 bind to. So protein or Proteus mirabilis were incubated with the recombinant LYPD8 protein, and then incubated with the secondary antibody, immunogold labeled secondary antibody that detects LYPD8, and then analyzed by the scanning electron microscopy. In this case, this area indicates the bacterial body, and this net-like structure indicates the flagella. And this immunogold particle can be observed at these flagellar regions, indicating that LYPD8 binds the flagella of the Proteus mirabilis. Then we analyze the motile activity of the Proteus mirabilis. Proteus mirabilis were inoculated at the center of the LV agar. In this case, this Proteus mirabilis expand over the LV agar. But in the presence of the LYPD8, this expansion was severely reduced, indicating that LYPD8 inhibits the motile activity of the gram-negative bacteria. So I would like to summarize this part. LYPD8 was highly expressed in the colonic epithelial cells and secreted into the intestinal lumen. And there, LYPD8 binds to the flagella of the highly motile gram-negative bacteria and inhibits their motile activity. And in the absence of the LYPD8, highly motile gram-negative bacteria invade into the colonic layers that increase the risk of the intestinal inflammation. Thus, LYPD8 is required for the separation of the microbiota and epithelial layers in the intestinal in the colon. So in the first part, I told that both the genetic predisposition and the environmental factor microbiota both contribute to the development of the intestinal inflammation. In the absence of the LYPD8, these animals became sensitive to the intestinal inflammation. But these LYPD8 knockout animals did not develop colitis spontaneously. So we wondered if microbiota composition was altering or not. So we performed the OTU analysis. Actually, in the LYPD8 knockout animals, microbiota composition was not altered, although some numbers of the gram-negative bacteria invade into the colonic layers. So we simply wondered if dysbiosis, altered the composition of the microbiota, induces the intestinal inflammation in LYPD8 knockout animals. So to address this, dysbiosis was induced by feeding high-fat diet. Mice were control diet or the high-fat diet, and then microbiota was analyzed. So high-fat diet fed mice, for example, the number of the lactobacillus bifidobacterium was severely reduced, and flagellated Escherichia was markedly increased. Thus, high-fat diet feeding successfully induced the dysbiosis, where the number of the flagellated bacteria was increased. So, wild-type animals and LYPD8 knockout animals were fed control diet or high-fat diet for 12 weeks. In this case, high-fat diet-fed LYPD knockout animals developed mild intestinal inflammation, as evidenced by the loss of the goblet cells or the invasion of the several inflammatory cells or the mucosal thickness. So, in the absence of the LYPD8, these animals became sensitive to the intestinal inflammation, but they do not develop the colitis spontaneously. Dysbiosis itself did not induce the colitis spontaneously. But in the absence of the LYPD8, dysbiosis contributes the spontaneous development of the intestinal inflammation, indicating that LYPD8 is actually important for the maintenance of the intestinal inflammation. Thus, intestinal microbiota are essentially present in the intestinal lumen without directly attaching the host cells. This is required for the maintenance of the intestinal homeostasis. But intestinal microbiota is now well known to influence the host cell functions. In this regard, bacterial metabolites, particularly short-chain fatty acid, are shown to act on the host cells, such as the epithelial cells or the immune cells through the G protein coupled receptors. In addition to the short-chain fatty acid, we wondered if other bacterial metabolites also act on the host cells. In this respect, we previously shown that bacteria-derived adenosine triphosphate, ATP, act on the immune cells to induce the Th17-mediated responses. So, ATP was shown to be derived from the commensal bacteria in the intestinal lumen. We so then wondered if how this luminal ATP is regulated. For this, we focused on the NTP-D-phosphohydrolase. This is the membrane-bound enzyme that hydrolyzes ATP, consisting of the 8 family members. Among these NPD8 family members, NPD1, also called as the CD39, was shown to regulate immune responses. So we wondered if other NPDAs regulates the intestinal inflammation. So Takashi first analyzed the expression pattern of these family members. And he found that NPD7 and NPD8 was highly expressed in the epithelial cells of the gastrointestinal tract. In the case of the NPD7, this NPD7 was highly expressed in the epithelial cells of the small intestine. And NPD8 was highly and selectively expressed in the epithelial cells of both the small intestine and the large intestine. So Takashi was first analyzed the role of NPD8 by generating the knockout animals. So in the NPD8 knockout animals, ATP concentration of the feces was not altered. But in the luminal contents of the small intestine, ATP concentration was markedly increased in this NPD7 knockout animals, indicating that this NPD7 regulates the ATP concentration in the small intestinal lumen. This is compatible with the expression pattern of the NPD7 in the small intestine. So we then analyzed the role of NPD8, actually Halka, together with the excellent Chinese postdoc, Bo Li. They analyzed the role of NPD8, which are expressed in the epithelial cells of the small intestine and the large intestine. And in the case of the NPD8, ATP concentration in the feces was markedly increased, actually indicating that NPD8 regulates the ATP concentration in the colonic lumen. This ATP concentration, increase of the ATP concentration was shown to be dependent on the microbiota because antibiotics treatment completely reduced the ATP concentration. So we then analyzed the increased, the effect of this increased concentration of the ATP for the regulation of the gut homeostasis. So mice were already treated with dextran sodium sulfate to induce the intestinal inflammation. Actually, NTPD8 knockout animals were highly sensitive to the intestinal inflammation. So NTPD8, which is expressed in the epithelial cells of the colonic and small intestinal epithelial cells, regulates the ATP concentration in the lumen. This ATP regulation of the ATP concentration is required for the maintenance of the intestinal homeostasis. In the absence of the NTPD8, ATP concentration was increased, and this ATP acts on the several immune cells to induce the intestinal inflammation. So we then analyzed whether other microbiota metabolites also regulate the gut homeostasis. In this regard, we focused on the unique subset of the intestinal myeloid cells characterized by the high expression of this CX3-CR1. These myeloid cells are present just beneath the epithelial cells and extend their dendrite into the intestinal lumen, directly uptake the luminal antigens, and subsequently induce the effector T cells. So I asked my PhD student Naoki to find out these bacterial metabolites that induce the dendrite protrusion of this unique subset of the myeloid cells. So he then prepared several fractions from the luminal contents of the small intestine of the SPF animals. So he actually prepared GHL ether soluble fraction, chloroform soluble fraction, and methanol soluble fractions, and used for the in vitro stimulation. So CX3-CR1 positive myeloid cells were isolated from the small intestine of these CX3-CR1 GFP animals and stimulated with these three fractions. In this case, methanol soluble fraction induced dendrite extension of these CX3-CR1 positive myeloid cells. So to further analyze the mechanism for the bacterial metabolite-dependent dendrite protrusion, we searched for the receptor, host receptor, that is responsible for the dendrite extension. Several previous reports have shown that G-protein coupled receptors are responsible for the recognition of the bacterial metabolites, particularly short-chain fatty acid. So we searched for the expression of the G-protein coupled receptors in this intestinal CX3-CR1 positive myeloid cells. So this R3 fraction, CX3-CR1 positive myeloid cells fractions, several G-protein coupled receptors are actually increased. Among these G-protein coupled receptors, GPR31 was most selectively and highly expressed in this CX3-CR1 positive myeloid cells. So we decided to analyze the role of GPR31 and generated knockout animals. So CX3-CR1 positive myeloid cells are isolated from the wild type animals and GPR31 knockout animals and stimulated with the methanol soluble fraction. So again, in the case of the wild type CX3-CR1 positive myeloid cells, methanol soluble fraction induced dendrite extension. But in the case of the GPR31 knockout cells, methanol soluble fraction did not induce dendrite protrusion. Thus, GPR31 was shown to be responsible for the dendrite extension of this methanol soluble fraction. So this is the case for the in vitro condition. So we then analyzed the in vivo status. So actually the 1 villus of the terminal ileum was analyzed by the 2-photon microscopy. So in the case of the wild type animals, dendrite extension protrusion was shown. But in the case of the GPR31 knockout animals, dendrite extension was almost gone. Thus, dendrite extension of the intestinal secretion positive myeloid cells was shown to be dependent on the microbiota and GPR31. So we now know that GPR31 is responsible for this dendrite extension. In the subsequent experiment, we tried to identify the bacterial metabolites that activates this GPR31. So by establishing the screening assay method to measure the GPR31 activation, we purify the GPR31 activating fraction step by step using several chromatographies. And these GPR31 activating fractions were finally analyzed by the mass cytometry. This mass spectrometry analysis have shown that these GPR31 activating fractions contain lactic acid. So we now know that lactic acid was contained in this GPR31 activating fractions. In the next experiment, we use the chemical compounds of this lactic acid. So we also use the chemical compound of the pyruvic acid, which chemical structure was clearly related to this lactic acid. So in the case of the wild type CXCR1 positive myeloid cells, lactic acid and pyruvic acid induced dendrite extension. But in the case of the GPR31 knockout cells, lactic acid, pyruvic acid did not induce the dendrite extension. This is the response in the in vitro conditions. We then analyzed the in vivo response. Wild type animals and GPR31 knockout animals were orally administered lactate or pyruvate for 3 weeks. And the terminal ileum, the villus of the terminal ileum was analyzed. So in the case of the GPR31 wild type mice, dendrite extension can be observed. And after lactate, or particularly for pyruvate treatment, dendrite extension was tremendously increased. But in the case of the GPR31 knockout condition, dendrite extension was not observed. Even after lactate or pyruvate treatment, dendrite extension was not induced. So we can say that bacterial metabolites, lactate and pyruvate in the intestinal lumen induces the dendrite extension of the CXCR1 positive myeloid cells through the GPR31. So microbiota derived metabolites mediates the host microbiota interaction for the maintenance of the intestinal homeostasis. So our next interest is whether microbiota derived metabolites are also implicated in the pathogenesis of the inflammatory bowel diseases. So Yuriko and Hisako together address this. So they first perform lipidomic analysis of the plasma of the Crohn's disease patients. So actually in the plasma of the Crohn's disease patient, several lipid metabolites were increased. Among these lipid metabolites, lysophosphatidyl lys-PS, lysophosphatidyl was most abundant in the Crohn's disease patient plasma. So this is a lipidomic analysis. We confirm whether lys-PS was actually increased in the plasma of the Crohn's disease patients. 2 kinds of the lys-PS was actually increased in the plasma of the Crohn's disease patients. We also analyzed the concentration of the lys-PS in the feces. Even in the feces, the concentration of the lys-PS was increased in the Crohn's disease patients. This lys-PS, lysophosphatidylserine, was generated from the phosphatidylserine by the action of the enzyme phospholipase A. And this phospholipase A is expressed in the host cells and bacterial cells. Because the lys-PS concentration was also increased in the fecal feces. We wondered if dysbiosis correlates with the increased lyso-PS concentration in the Crohn's disease patients. So for this, we performed shotgun sequencing of the feces of the healthy individuals and Crohn's disease patients. So actually, dysbiosis can be observed in the Crohn's disease patients. Because this is a shotgun sequence, we can precisely analyze the presence of the genes, particularly phospholipase A genes. Several bacteria have the phospholipase A genes. Among these bacterial phospholipase A genes, ECSF3660 was really increased in the Crohn's disease patients. This ECSF3660 was mainly derived from the Escherichia. So in the Crohn's disease patient, they have the dysbiosis with the increased number of the microbiota possessing the phospholipase A genes and increased concentration of the lysophosphatidyl serine. So our next interest is whether this lyso-PS is implicated in the intestinal inflammation. So to address this question, we use the mouse colitis models. We first introduced TNBS colitis models, mouse models. Mice were immunized with the TNBS on their back skin. And 1 week after the immunization, TNBS was administered to the via the transanal route. And at that time, lyso-PS was also injected. Lyso-PS treatment induced severe intestinal inflammation, and they showed severe weight loss. Thus, lyso-PS treatment exacerbated TNBS-induced intestinal inflammation. We also introduced another colitis model, T-cell dependent colitis model. LAG2 knockout animals, immunocompromised animals, were transferred with the naive T cells to induce the T-cell dependent intestinal inflammation. And 17 days after the naive T cell transfer, lyso-PS was administered. So lyso-PS administration was induced really severe, rapidly induced severe intestinal inflammation, and they showed severe weight loss. In these animals, we then analyzed the number of the T cells in the lamina or colonic lamina propria. So actually, in the lyso-PS treated animals, the number of the interferon gamma producing Th1 cells, interferon gamma and IL-17 double producing pathogenic Th1 cells were increased in the colonic lamina propria. So lyso-PS treatment exacerbated the intestinal inflammation in 2 types of the T-cell dependent colitis models, together with the increased number of the Th1 cells. So we then analyzed whether lyso-PS directly acts on the CD4 T cells or the other types of the immune cells. So we purified naive T cells from the spleen, and these naive T cells were cultured under the Th1 polarizing conditions. And 24 hours after the culture, lyso-PS was added to the culture. In this case, addition of the lyso-PS increased the production of the interferon gamma, indicating that lyso-PS was actually directly acting on the Th1 cells to activate the Th1 cells. Because when naive T cells were stimulated with lyso-PS, in this condition, the naive T cells was not activated. Thus, lyso-PS was shown to directly act on the Th1 cells to activate the Th1 cells. So we then analyzed how lyso-PS act on these Th1 cells by identifying the lyso-PS receptors. So in the case of the lyso-PS, several host receptors was already shown, such as GPR34, P2Y10, and GPR 174. So we analyzed the expression of these lyso-PS receptor in Th1 cells and found that P2Y10 was really highly expressed in the Th1 cells. So we generated P2Y10 knockout animals and introduced into the LAG2 knockout dependent T-cell colitis models. So LAG2 knockout animals were transferred with naive T cells of the wild type animals and P2Y10 knockout animals. So the transfer of the wild type naive T cells induced mild intestinal inflammation, and they showed mild weight loss. But the transfer of the naive T cells from the P2Y10 knockout animals did not induce anything. And in this case, 20 days after the naive T cell transfer, at this time point, lyso-PS was administered. The lyso-PS administration induced the rapid, so actually lyso-PS treatment induced severe intestinal inflammation very rapidly, and they suffered from this very severe weight loss. But in the case of the LAG2 knockout animals transferred with the P2Y10 knockout naive T cells, lyso-PS treatment did not induce anything, indicating that lyso-PS acts on the Th1 cells through the P2Y10. So I'd like to summarize this part. In the Crohn's disease patient, they have the dysbiosis with the increased number of the microbiota possessing the phospholipase A genes. And they showed the increased concentration of lyso-PS. And this lyso-PS act on the Th1 cells through the P2Y10 to directly activate the Th1 cells and worsening the intestinal inflammation. So I'd like to finish in this part. So I'd like to acknowledge the contributors to this presentation. On the first LYPD8 part, Liu mainly performed the experiment, and the part of the GPR31, Naoki mainly performed the experiment. And the final part, Hisako, together with the Yuriko, performed the experiment. So thank you very much for the hearing.